This is Jeffrey Borer. I'm talking about radionuclide-based imaging of the heart. Radioisotopes, or radionuclides, administered into the blood via peripheral veins can enable imaging of the heart. From these kinds of studies, cardiac geometry, chamber function, myocardial perfusion, and even myocardial metabolism and other aspects of cellular and molecular biology can be determined. These techniques have been widely used for clinical evaluation and form the basis of management decisions in many common cardiac diseases, and most particularly in the area of coronary artery disease, though they've been used in patients with valvular disease and cardiomyopathies as well. Some of the techniques, particularly those that interrogate myocardial metabolism, may be of clinical utility, but are primarily of interest for research purposes into the pathophysiology of cardiac dysfunction in a variety of disease states. Now, the most commonly used radionuclide-based technique is myocardial perfusion scintigraphy. Like other radionuclide-based techniques, this is relatively non-invasive because the only entry into the body required for performance of the test is the peripheral venous injection of an isotope. Myocardial perfusion scintigraphy involves the peripheral venous administration of a radionuclide-based perfusion agent, either thallium-201 in the form of thallus chloride, which is handled sort of like potassium by the myocardium, or any one of several technetium-based agents. These imaging agents distribute within the myocardium primarily according to coronary blood flow. Photon emission data are collected with a special radiation detector, which comprises a sodium iodide crystal with some electronics and photomultiplier tubes behind it, so that photons that impinge on the detector can be localized in space as to their likely source of origin. In order to localize them, the assumption is made that the photons strike the detector face only if they impinge on the detector from a direction that's perpendicular to the plane of the detector face. To ensure that that is true, various collimators, like lenses, are used in front of the detector, which preclude photons that aren't oriented basically in a direction perpendicular to the detector face from hitting the detector. Given the use of the collimator and the assumptions that can be made because a collimator is used, it's possible to collect radioactive emissions in a spatially organized way as they hit the radiation detector, and to use that information to reconstruct in a computer the spatially organized display of the radioactive emissions, which can give you a picture of the heart, either of the myocardium itself, as in the case of myocardial perfusion scintigraphy, which we're now discussing, or the blood pool, if it is labeled, as in radionuclide cineangiography, that I'll discuss in a few moments. Now, with myocardial perfusion scintigraphy, radionuclide distribution within the myocardium remains constant for many minutes after the isotope is administered. Therefore, the perfusion agent can be administered during exercise, and imaging can be performed a few minutes later when patient motion required for exercise no longer is a consideration, so that the quality of the image can be better with the patient at rest. It's possible, therefore, to use a radiation detector that revolves around the patient during imaging, because the patient can be imaged while he is at rest, and this enables collection of images from many different angles. Computer-based reconstruction of these data permits generation of multiple two-dimensional slices along any cardiac axis. This effectively provides three-dimensional data about photon localization and intensity. This technique is called single photon emission computerized tomography, or SPECT. Coronary artery obstructions are detected when the perfusion agent fails to localize in any myocardial region. Relative maldistribution of coronary flow is potentiated by increasing the myocardial oxygen demand, causing increased flow to normal regions, and relatively or absolutely decreased flow to regions supplied by partially or totally obstructed arteries. Therefore, detection of hemodynamically important coronary artery disease is most efficient when both rest and post-exercise images can be obtained and compared. Distribution of isotope within pulmonary vessels can support the inference of pulmonary vascular congestion during exercise, but the clinical utility of this observation is relatively modest compared to the information obtained from the myocardial perfusion study itself. Myocardial perfusion scintigrams can be used not only to detect the presence of coronary artery disease, but to provide useful information about prognosis in patients with ischemic heart disease. This is true because the size and intensity of perfusion defects and the multiplicity of perfusion defects and the total area of myocardium affected by perfusion defects all are measures of the functional severity of ischemia and therefore of the prognostic importance of coronary artery obstruction. And the tests are used in this way commonly. So myocardial perfusion scintigraphy is useful for identifying coronary disease and also for determining prognosis. Perfusion scintigraphy also can be performed with high-energy radioisotopes prepared in a cyclotron, so-called positron-emitting radionuclides. These isotopes produce two photons emitted at equal intensity and precisely in 180 degrees opposition. Radiation detectors similar to those that are used for myocardial perfusion scintigraphy are used for detecting these isotopic emissions, but the crystals need to be thicker than those used for myocardial perfusion scintigraphy because they have to capture very high energy photons produced by these positron emitters. The thick crystals are placed in a ring around the patient so that both photons can be captured with each emission. This is called coincidence counting. The availability of these two spatial cues for each emitted positron enhances the capacity for spatial localization, and the time at which each of the two diametrically opposed photons impacts on its detector also can enable improved spatial precision in reconstructing images. This technique is called positron emission tomography, or PET, and it can be used for perfusion scintigraphy, but it has a wider utility that has found some application in the area of coronary disease in clinical use and some application in research in other areas like valvular disease and cardiomyopathy. Almost any metabolically active molecule can be labeled in a cyclotron with a positron-emitting atom, and therefore PET can be used to track metabolic pathways within the myocardium or to define the localization and fate of molecules involved in specific intracellular or even intranuclear reactions so that gene expression can be determined with PET when appropriately labeled reporter genes are transfected into tissues of interest. This technology for clinical purposes is used primarily in patients with coronary disease to determine regions of anaerobic metabolism So it's possible to use, for example, fluorodeoxyglucose and labeled fatty acids to determine which regions of the heart are metabolizing in a normal fashion and which aren't, and to make inferences about the existence and perhaps severity of ischemia and the matching of flow and metabolism in various areas. This can be very useful in defining whether specific regions of tissue are ischemic but still viable, and that's important in making certain specific decisions about revascularization, where a graft is likely to do some good and where it may not, or where an angioplasty will lead to recovery of function and where it won't. In the setting of patients with ischemic heart disease and heart failure, where recovery of muscle function is particularly important, positron emission tomography can be helpful in defining whether or not a patient is likely to benefit clinically from a revascularization procedure. Now, what I've defined so far is imaging focused on the heart muscle itself. And there are other techniques also that focus on the heart muscle. Labeled antibodies can detect damage to myocardium. For example, anti-myosin antibody can detect the presence of a myocardial infarction by identifying free myosin in an area of myocardium that shouldn't be there. It should all be intracellular, and the isotope shouldn't be able to get in. It's also possible to use radio-labeled ligands of other sorts to detect certain other kinds of metabolic abnormalities or to trace metabolic reactions in the heart. However, for our purposes, most of these are of research interest and have limited clinical applicability and most particularly have limited clinical application in the United States. However, in addition to myocardial imaging, there's another whole area of imaging that's of radionuclide-based imaging that's important clinically, and that is blood pool scintigraphy. With this technique, the radioisotope is attached to the red blood cells so that one can follow its movement and the movement of blood through the circulation, or more particularly into and out of specific cardiac chambers. The radionuclide-based method that's most commonly employed for this purpose is called radionuclide cineangiography, or radionuclide ventriculography, or blood pool scintigraphy, or radionuclide angiography. They're all the same thing. They're performed, this technique is performed with the isotope either injected as a bolus and followed through the heart, that's called the first pass technique, or more commonly, once the isotope has thoroughly mixed in the intravascular blood pool and therefore is at equilibrium. This technique then allows for the visualization of the cyclic variation in blood volume in each cardiac chamber. Because the isotope is at equilibrium in the intravascular blood pool, radioactive emissions that are detected from the blood pool with the sodium iodide crystal placed over the chest can be related to the amount of blood from which the isotope has been emitted, so that it's possible to get fairly precise measures of relative and absolute intra-chamber blood volumes when radionuclide cineangiography is performed. This technique requires a peripheral venous injection of a small amount of technetium-99m, usually 10 to 30 millicuries of technetium-99m, in a form such that it will interact with the ligand, stannous pyrophosphate, that binds the radioisotope to the circulating blood cells for many hours. Once the technetium has equilibrated in the blood pool, a sodium iodide radiation detector is placed over the patient's chest. Again, a collimator is used so that precise inferences can be made about the specific site of origin in physical space of all the photons that strike the radiation detector crystal. and therefore, once the information is transmitted via photomultiplier tubes to the appropriate circuitry and to a computer, it's possible to organize the images into pictures of the location of blood within the heart during any instant throughout the cardiac cycle. In order to make these images most interpretable, multiple images need to be obtained with sufficient temporal resolution to provide an accurate reflection of the variation in intra-chamber blood volumes throughout the cardiac cycle. And for this purpose, the computer receives an additional signal from what's called a gating device that identifies the onset of each heartbeat. Usually the QRS complex of the electrocardiogram provides this gating signal. The computer then collects the data in temporally organized units or movie frames that span the entire cardiac cycle and can be displayed for visual analysis as an endless loop movie. The temporal resolution of the system is determined by the computer algorithm that's used, but it has to be sufficient so that major functional parameters are not importantly affected by frame duration. In other words, it's important that there be enough frames that are taken quickly enough so that you don't sort of suppress the peak of the ejection fraction, or the minimum or the end systolic volume is sort of made to look bigger than it really is by diluting with volumes from earlier in systole or later in diastole. For most purposes, it's necessary to have a temporal resolution of at least 40 milliseconds. In other words, the frame can't be longer than 40 milliseconds for most of the heart rates that are achieved during exercise, although sometimes you need to get down to a 10 millisecond temporal resolution for certain functional assessments. You need about 2 million photons in an image series to ensure the capacity to differentiate efficiently between intracardiac blood and background emissions from the lungs, the chest vessels, and what have you for accurate image interpretation. Any single cardiac cycle comprises an image series with far fewer counts than this, and so the computer collects temporally organized frames throughout many cardiac cycles during an interval spanning at least 2 minutes, and then superimposes corresponding frames digitally so that the resulting movie is a composite of the average heartbeat during the period of imaging. In the most highly developed imaging systems, each photon receives 3 digital addresses in the computer memory. One defines its spatial location in X and Y coordinates. The second defines the clock time of its arrival, and the third indicates the heart rate at the time the photon was collected. This is called list mode data acquisition and allows the interpreter to select the movie corresponding to any specific heart rate and therefore to eliminate arrhythmic beats and other problems during imaging. The results of radionuclide cineangiography, which is really totally analogous to contrast angiography, is that allows you to define functional parameters quantitatively. So you can define the left ventricular ejection fraction, the right ventricular ejection fraction, ejection rates, filling rates, absolute left ventricular volume, absolute right ventricular volume, etc., etc. This kind of information is very useful clinically because the change in left ventricular ejection fraction from rest to exercise is a primary measure of the functional severity of ischemia in patients with coronary artery disease, and also is a measure of the state of the myocardium in a variety of other cardiac disorders, specifically in regurgitant valvular diseases, and most particularly in aortic regurgitation. This kind of information provides probably the best prognostic information that we can obtain. It's not radionuclide cineangiography is not used as frequently as it once was in patients with coronary disease because it's possible to obtain an ejection fraction from a myocardial perfusion scintigram as well, and the ejection fraction obtained during exercise from the myocardial, or immediately after exercise from myocardial perfusion scintigram, provides useful prognostic information which can be added to the information from the scintigram itself. But if you want the most precise ejection fraction or the most precise prognostic data, obtaining blood pool scintograms and determining determining ejection fraction at rest and during peak exercise is particularly useful. And in fact, to the best of my knowledge, the only data that define the relative risk of operating and not operating in patients with severe coronary disease is based on radionuclide angiography. The technique also is very useful in identifying patients who are asymptomatic with valvular heart disease, and particularly with regurgitant valvular diseases, whose risk is sufficient so that it is reasonable to consider prophylactic operation despite the lack of symptoms. As I mentioned earlier, there are a variety of other radionuclide-based techniques that can be employed to ask particular questions about the heart and about cardiovascular pathophysiology in a variety of disease states, but these are primarily of research interest. It is possible to interrogate the density of adrenergic receptors, for example. It's possible to image atherosclerotic plaque to a greater or lesser extent with certain experimental imaging agents. There are a number of questions that can be asked. These techniques will be used more as research goes on because our knowledge of cardiac pathophysiology is entering a cellular and molecular phase now, and the use of radiolabeled ligands to interrogate cell biology and molecular biology makes these techniques particularly attractive as we start thinking about how to make decisions on a more fundamental level than we have in the past. Thank you very much.